Good morning, viewer. Biendas here. Yes, the most amazing news for our viewer. Yeah, fighting cancer effectively often involves stopping cancer cells from multiplying. Yeah, which requires understanding proteins that the cells rely on to survive. Protein profiling plays a critical role in this process by helping researchers identify proteins and their specific roles that future drugs should target. But when used on their own, past approaches have not been detailed enough to spotlight all potential protein targets, leading to some being missed. Now, by combining two methods of protein analysis, a team of chemists at Scripps Research has mapped more than 300 small molecule reactive cancer proteins as well as their small molecule binding source, revealing key protein targets that when disrupted with certain chemical compounds or small molecules halt cancer cell growth may eventually enable the development of more effective and precise cancer treatments. The findings appear in Nature Chemistry. One method gave us a broad view of which proteins were interacting with the chemicals, and the second method showed exactly where those interactions were happening, says senior author Benjamin Cravett, PhD, the Norton B. Gilula Chair in Biology and Chemistry at Scripps Research. Both methods are forms of activity-based protein profiling, a technique that Cravett pioneered to capture protein activity on a global scale. The research team used their dual approach to flag both the proteins and protein seeds that interacted with a library of stereoprobes. Yeah, chemical compounds designed to permanently bind to proteins in a selective manner. Viewer, stereoprobes are used to study protein functions and identify possible drug targets. We made a conscious effort to design our stereoprobes with chemical features that tend to be underrepresented in compounds typically used in drug discovery settings, says co-senior author Bruno Malio, PhD and institute investigator in the Department of Chemistry at Scripps Research. Yeah, this strategy increases our chances of making discoveries that can advance biology and eventually translate into improvements to human health too. Biendas exclusive report.